Good morning, everyone. I am welcome you all to this 8th day of CSIR question and answer discussion session. Today we are going to discuss about the unit 11 and 12, the questions. Before entering into the session, please let me know, am I audible? Can you see my slides? Is it visible? Please type it in chat box. Am I audible? Good morning, everyone. Am I audible? Can you please type it in chat box? I'm not sure about it. Yeah. Okay. Thank you, everyone. Thank you, Kavan, Yasmin. Thank you, Yasmin. Okay, we'll start this sessions. Okay, today our question and discussion is fully related to the topic called the evolution and behaviors, then applied biology in unit 11, and this applied biology in unit 12. In this question, there are some year of questions they are asking the unit 12 questions. In some questions they are asking unit 11 questions. So in this today class fully about the mixed session of the both unit. Here the first question we are going to discuss here is recent studies on archaea suggest that life could have originated. They are given a statement that called the archaea suggested that life could have a originated from which, that is the question. Here the answer is in deep hydrothermal vent. The deep hydrothermal vent means that hydro means that is the water, thermal is a temperature. At a temperature change in a water, that makes the life starting. Whenever we are studying the evolutionary studies, that living organisms are getting started from the water. That as per the scientific report, are used from the start their life from the water. So temperature changes in the water makes the new organisms, so originated from the deep hydrothermal vent. That is the answer here. And next question, if the ratio of number of non-synonyms to synonym substitutions. Here the statement is ratio of non-synonyms and synonym substitutions per site in a protein coding gene is greater than 1. Okay, protein coding gene means that there are n number of protein coding genes in a chromosome, in our genetic material DNA, in our genetic construction. In that they are asked the question, it is an evidence of selection, that is the how can we get whether it is passed. So here they mention that protein coding gene is greater than 1. And another thing, the ratio of non-synonymous and synonymous substitution they provided, the ratio is greater than 1 means we can tell that is the positive. If the ratio of the synonymous and non-synonymous substitutions are being less than 1, that is the point value, means that is the negative value, that evidence selection that is negative. So here the two things we want to understand. If it is greater than 1, that is positive, less than 1 it is negative. So the ratio become equal, that are 0, that 0 not 0, that is equal. Both are equal means that is neutral. These are all the things we want to have in this part of and the question, this kind of question. Then next question, the frequency of allele A, capital A and small a. The frequency of capital allele capital A and capital A and small a in a population at Hardy Weinberg equilibrium. The thing statement here is Hardy Weinberg equilibrium are 0.7 and 0.3 respectively. So allele A has the that equilibrium called 0.7, small a having small the 0.3. Random sample 250 individuals taken from the population. How many of are expected to heterozygous? So here they are taken the group of sample. There are 250 peoples are there. In that 250 peoples, they are asked the question expected heterozygous. Heterozygous means here there are that people are not from same species. They are from various species. That is hetero. Homo means that homozygous means that both are comes from same origin. This kind of questions are asked the evolutionary calculation with the equation called the Hardy Weinberg equilibrium. In this equilibrium that yesterday okay, I will send the material. Today I will send the material. There are the derivations in the equations. In the equational part, they have some calculations with the 1 and 0.3 values. In equation, we want to find that 0.7 and 0.3 totally there is one value. So total is called as 1, that is the 250. In the 250 0.7, that ask the heterologous. In the heterologous is the that both are there is a possibility called 0.7 possibility is also there, and heterologous, that is the product is in the form of addition of capital A and small a. This like this we want to do this equation. I will send the equation in the today material, and that answer is become 105. This comes from this Hardy Weinberg equation calculation. Next part of this question is the altruistic act. If a donor sacrifices C offspring, which helps the recipient to gain B offspring, and the donor is related to the recipient by coefficient gamma, and under which condition ever that altruistic trial. In this we want to correlate the answer. How can we correlate a donor? If the donor sacrificing C offspring, so that C offspring is a C offspring, and that helps to recipient B. Recipient B means that is the B is a +1, so we want to take a B. That donor related to the that recipient by coefficient called as the that recipient having the gamma. So this gamma value is present with the recipient by the coefficient gamma, and the condition under which condition kin selection favors this kin selection theory. They are mentioned this kind of the traits are having more than 0 possibilities, that is that the offspring protection is higher than the 0. Then okay, the photoreceptor commonly involved in a light entrainment of mounds and plants is which kind? It comes under which? how they are utilized. This photoreceptors are commonly involved. Photoreceptors means there are four type of answers are here: phytochromes, carotenoids, and cytochromes. In this four, they are asked light entrainment of biological clock. Which compound is it's helpful for their light entrainment? Means that is the cytochrome. The cytochromes are it's present in the plant that are depends on the light from itself. We are having the two different type. That two different type, that is phytochrome A, that is light stable and light sensitive. That light stable molecules, cryptochromes are involved in the that they are involved in the daytime. That's light stable cryptochromes are involved in the light sensitive materials are involved the biological function of the plant in the the dark condition. So that functions, the molecules are changed, their shift based on their functional activity. So in daytime, that molecules that structure, the amino acid domains in structure become vary in between the day and night. So this day and night make the clock of the particular plant. Then next about the concept is which of the following food crop has recently been genetically engineered to obtain edible vaccine to develop immunity against hepatitis B. Hepatitis B, there are some food crops are genetically modified because it's the latest research related update. This is the B that vaccines are developed in a crop called potato. Then release of nutrients, oxidants, electron donors into environment is to stimulate naturally occurring microorganism to degrade the contaminant. This here the release of some microorganism, that micronutrients, oxidants, electrons into the environment. It's stimulating the naturally occurring microbes. Here the stimulation happen. The one thing here want to mention, the key word is the environmental stimulation. We are all know microorganism which involved in the contaminant degradation of contaminants is called as the bioremediation. So they mention the key word here, that environment stimulate the microorganism. That normal microorganism that can't degrade the contaminant. That microorganism needs some nutrients, oxidants, and electrons for their special character of degradation. So that stimulation process is called as biostimulation. Okay, next is a very direct question. ELISA reader. The ELISA assay uses. ELISA assay is a very familiar and very widely used method in that industry and medical field. That is a substrate which gets converted into the colored product. In ELISA, the colored product formation which helps to understand the amount of reaction happened. That reaction is measured based on their color intensity. That colored intensity which helps to understand, okay, this much amount of substrates are converted, so this protein is in active form. So this ELISA helps to understand, it uses a substrate which gets converted into colored product. Here the confusable answers here, because we are all know ELISA is fully about the enzyme linked compounds. This enzyme linked compounds are having the activity that called here they provided the enzyme which gets react with secondary antibody. All those things are it's about something about the ELISA, but not the perfect match. Here the perfect match, The final step of is that utilizing the substrate and producing color. That is the final best answer. Whenever you are seeing the similar answer, find out the which is the best one, which is the exact understandable. Then routinely used glucose biosensor estimated blood glucose level by sensing the concentration. So it's very direct. Biosensor estimation, that glucose biosensor estimates the blood glucose level sensing the concentration of oxygen. Okay, the concentration of oxygen in the blood, that makes a point. That's the points shown in the display. That points are fully depends on the that oxygen concentration in the blood. If that's oxygen it's in higher quantity, we can easily get this in a larger quantity. Okay, the next part of question. Okay, first living being on earth were anaerobic. There are anaerobic. Why? Because there is no oxygen in the air. It's a statement which given by the Darwin's. This statement is well studied and proved concept. The first living being on earth were anaerobic, that is comes from under the learned. That learned parts are divided, and that part filled up with water. That organism which utilizing the oxygen which present in the water, then that is make conversion on their life cycle. So first living being on the earth were anaerobic because why? Because there was no oxygen in the air, so there are dumped in the land. Landfills are the land. One normal land is the first source of the microbe, which to keep them here. The next question, to keep them in a totipotent state, embryonic stem cells need to maintain stem cell. Embryonic, here the main concept keyword is a totipotent state, that embryonic stem cells needs to maintain the organ in a one particular condition, that is not enlarge, not develop, because there is a no no blood vessels are formed in the cells. That inhibiting factor helps to inhibit all those functions of the protein, the functions of cell. This is very important concept in the latest research, because whenever they are studying the cell culture study, that we want to understand the concept in the growth hormones, insulins, and other additional supplements. Okay, these other additional supplements are playing vital role in the functions of all those the cell development and totipotent statement. All those stages are fully relevant to the concept called the growth animal cell culturing. Okay, this animal cell culturing related studies are its latest trending methods. And then next concept, the peacock tail is example for. Okay, the tail here they mentioned peacock tail. Female peacocks don't having their tails, but the male having their tails, but the male having the tail. So the sexual grouping selection. Then next thing is in the sexual selection result is a variation in the reproductive success of male. This is the statement often due to the female choice with particular phenotype. This type of sexual selections occur because the host of breeding is higher for female as compared to the male. So this the cost is higher in this type of selections is become higher. In here they provided the two statements. The statements, the selection, the sexual selection, and the variation in the reproductive success of males. The variation is success. Its variation in the reproductive success of males is due to their female choice with particular to phenotype. So that is the cost of breeding is higher of for female as compared to the male. It's very next question is very easiest question. Among the following event in the history of life, that there are in the history of life, we are there are some events, sequential events are happen. What is that events? First thing, the organic molecules are produced. Okay, the organic molecules are produced in a history, then that organic molecules are become self-replicated. That's a self-replicated molecules undergone natural selection. After the natural selection, the prokaryotic cells are formed. After the prokaryotic cells formation, the eukaryotic cells are that are produced. These are all the order of the life history of life. Then the Hardy-Weinberg principle. This is very Weinberg is very important in this evolutionary study. It comes from considering what happened when the Mendelian gene act in the population. The model predicted that there will be no change in allele frequency. This is the statement that we want to tell the solution for this statement. There is no evolutionary process is at work. There is no evolutionary process in the working stage. And which of the following is responsible for maternal behavior in the first time pregnant rat after the parturition? Okay, in this here the statement they are provided is the okay, the responsible for initiation of the maternal behavior. The maternal behavior means that is the that initial parental behavior in that first time pregnant rat. In this rat, they mention after the parturitions. In this stage, the higher prolactin level in the blood. So there is a that initial maternal behaviors happen in the rat, first time pregnant rat. The next question is that okay, replace animal use in the testing hepatic toxicity of drug on trial, which one go into be closest to in vivo scenario. This is which is the similar content, similar level, that liver slices are the place that replaced animal is the similar to replace animal use in a testing hepatic toxicity of drug trial, which would be the similar in vitro and in vivo. Means there are liver cells are the hepatic cytotoxicity is a trial. So here in the answer there is a confusable answer there. The hepatic cell lines, we don't select the we can't select the hepatic cells. We want to select the our liver slices, which only shown in vitro and in vivo. Which of the following does not represent a strategy of phytoremediation? Here the mentioned not represent the phytoremediation. Phytoremediation means that some chemicals or materials are degraded by the end of question here. Here the which is not a strategy. Here we want to understand the four concepts. The phytodegradation. That phytodegradation is a normal that roots of plant are releasing some compounds. That compound degrades the that material that is by the plant. That is a biodegradation. Sorry, phytodegradation. Phytomining, that is the breaking down of material, and continuous removal through the hyperaccumulation. Hyperaccumulation, we all know that plant utilizing the chemical, that chemical is present in the plant leaf or stem. We can recover that for some accumulating period. Then chelate mediated extraction of pollutant, that we want to that filter the chemicals from air that stored in their surface material, that surface of plant, or they are utilizing for their growth, or and they are producing some chemicals. So there, the thing here, the degradation is not happen in the phytomining. And word fermentation is used in biochemistry. Sorry. The word fermentation is used in the biochemistry and microbiology denote different phenomena. The form it if the former is called C and latter is called T. The former is called C, later is called as the T. Which of the following statements is easiest, but somewhat they are confuses. Don't confuse with this statement. Please follow the first statement. Fermentation is used in a biochemistry. That is the first one in microbiology technique. So denoted the different phenomena. The use of here the enzyme formers is the previous one, latest is the advanced one, CNT. All C is T, but all T is not a C. Here the next is all T is in C, but all C's is not T. And the third option, T is originating while C is not. And second question, C is always anaerobic process while T is a aerobic process. So first thing of the fermentation in a microbiology technique that happen in an anaerobic process. So similar thing, that former things happen in an anaerobic condition. Later the process become anaerobic or aerobic, that is the advanced method. Then which of the following statements is not true to agrobacterium? This agrobacterium is a plant pathogen which helpful to prevent that are helpful to that they are agrobacterium tumefaciens are they are not helped in the field, they are helps to in the research laboratory to develop the recombinant plants. This Acrobacter tuberculosis, which is not true. So the not true statement is a D, a correct answer. So we want to go through. The protein product of virulent gene Vir A and Vir G preserves the acetostringons. So here this may be the question in the next your CSIR question exam. So please listen. The protein product of virulent gene Vir A and Vir G precise the X. The Vir protein forms a connection between Acrobacterium and the plant cell and facilitate the tDNA transfer in a plant. It's make a joining between the Vir B, make a connection between it's helpful for the tDNA transfer into plant. tDNA is containing the virulent gene, that is the vector, that is in this concept, the Vir B concept, this concept is helpful for the production of recombinant plants. The tDNA is modified, we can modify this tDNA. This tDNA is existed. Okay, these are all the corrected answer, which is not true. This fourth statement is not about the agrobacterium. The tDNA, after becoming coated with Vir F binding to the phosphorylated VIPI, which allows the complex to enter the plant's nucleus. So this is not involved after the nucleus level. Then next concept is the character similarity that can be misinterpreted. The character similarity is misinterpreted as common distance is called. Here the thing is the character similarity misinterpreted here the character similarity with G, and that means the simply synonymous and synonymous. These are all the that meaning of explanation of the character similarity, which is misinterpreted means homoplasy. That homoplasy is not about the character similarity. Which of the following evolutionary process played an important role in evolution of complex immune system? Which of the following evolutionary process play a important role in a that complex evolution is a co-evolution. In some species of New World monkey is advanced one. Only one female produced in a group. One or more younger females have suppressed reproduction and assisted the reproductive females. This example for which selection? This is called kin selection. Some world monkey, there is only one female produced in a group. That few younger females are suppressed reproduction, that are assisted the reproduction, they need assistance. So this kind of group, this example for the kin selection. Then bird species were both parent contributed equally to the parental care. This parental care is here they mention the bird species are both parents contributed equal parental care. So this both sex are morphologically similar, so only they are care each other. The next part is idea that altruistic gene, it that means this kind of genes are the that makes a similar function and similar activity. The similarity that is mentioned as the allustris means that is the that cell that is not they are not that is in the same family, but they are not they are doing the same activity, same similarity appearance, but that are not correlate. How can I tell that behavior is okay, 1 minute wait, I'll let you know. That is that difference between that individual that are having the that individuality is become they are not work for themselves. If for example they that if the organism species or gene that needs to be do one function means that not able to do that function, that needs the assistance of other protein, that kind of proteins are here. But this kind of genes are similarly present in all over this all organisms will be favored if R greater than C by B, where R is coefficient of relatedness, B is benefit of recipient occurred. Cost occurred means that is the that energy utilized, that utilized energy rate. Okay, that they help someone who who have a need. So that at the if the cost occurrence is the happen with the animal or gene. Here the answer is the Hamilton's rule. As per the Hamilton's rule, the this statement is reported by B. The use of double haploid in a plant breeding helps to reduce generation time and while intercrossing the recessive traits. The next question, the sustained successive generation of cell line in a culture. The ideal genes transfer can be obtained using the lentiviral vector. This is the method which lentiviral method is advanced research technology which in many laboratories are worked on with on this lentiviral. The sustained expression of transgene is the that expression which happen throughout their life cycle. The transient successive generation means that is